Macromolecules. Macromolecules are composition of larger component to form macromolecules using polymerization. These smaller components are called the monomers, and the constituent of this macromolecule include the carbohydrate, lipid, protein, and nucleic acid. There are 4 types of these macromolecules that we can use or modify to conduct advanced microbial tests, and these include carbohydrate, protein, lipid, and nucleic acid. Nucleic acid composed of the RNA and the DNA constituent. Carbohydrate. Carbohydrate composed of carbon, hydrogen, and oxygen monomers that comes together to form the carbohydrate. And there are 2 categories of this carbohydrate: the simple and complex. The simple composed of the monosaccharide and the saccharide. Under the saccharide we have glucose, 6 carbon sugar, fructose, 5 carbon sugar, and galactose. Under the saccharide we have maltose, lactose, and sucrose. Then protein. Protein utilizes amino acid monomers to form protein. They are jointly together with carboxylic and amino group, and there will be no lipid or carbohydrate without protein because the enzyme used for their synthesis are protein themselves. From the depiction you can see several types or structure of protein. And lipid. Lipid utilize hydrophobic set of macromolecules as monomers to form lipid. They do not dissolve in water. This involves triglyceride, catechol, phospholipid, and steroid. They help in the formation of the cell wall or cell membrane, and they can also form several hormones. From the picture you can see several types of lipid and their formation, from the hydrophobic group to non-polar hydrophobic group. Together they form constituent of the lipid as glyceride. For a nucleic acid, as they consist of the DNA and the RNA polymers, and together they form the nucleotide sequences. The DNA stands for the deoxyribonucleic acid, which is double-stranded, and the sequences are paired between adenine and thymine, then guanine and cytosine. While RNA stands for ribonucleic acid, and the sequences are paired between cytosine and adenine, then guanine and uracil. That means there is difference between DNA and RNA by their guanine and uracil component.